3 waiting. Yeah, I get it. That is it. Oh, there is a lag period between the two, right? Shall I start?
Yes, sir. Can I start?
Okay, good afternoon, dear students. Am I audible? NPTEL, can you once confirm whether I'm audible? Yeah,
yes, sir.
Thank you.
You are audible, sir. You are audible,
Thank you.
sir.
Thank you. I welcome you to the live session number 1 on genome editing and engineering. Sorry for the interruption, because I was also opening the YouTube, so there was a echo. So let me restart. I once again welcome you all to the live session number 1 on the course Genome Editing and Engineering. So currently I'm trying to check how many students have joined. Not many students in online, but some people are watching there in YouTube. Anyway, so let me, if there are some queries, kindly type so that I can address them. Otherwise, let me slowly start with some of the questions that were asked by some of the students. So number 1, there was a query from Madhu Ammu 600@gmail.com. Full details of the student is not available. So he has a very she has a very simple question. She was asking whether what is happening in this video at 7.35, and the question is how to describe a DNA is a transforming principle. So basically in this lecture, we were referring to Griffith's experiment, where he had taken a rough strain which is non-virulent, or so if this rough strain of Streptococcus pneumoniae bacteria is injected into mice, as this is non-virulent and safe, it will not cause any disease, due to which the mice will live. Okay, mouse will not die. The smooth strain which is the virulent strain and causes disease and also fatality, if injected, as in the second case, will lead to death in the mice. So here you can see a rough strain, which is safe and do not kill, and there is a smooth strain, which is virulent and causes fatality. Now, if we take the rough strain and heat-killed smooth strain, since we have killed the smooth strain by heating, what would happen? That is number one case. And second case is if we take heat-killed smooth strain and inject it, then also the mouse will be alive. So injecting a heat-killed smooth strain and injecting rough strain gives us the similar result where the mouse do not die. But if we, so both the strains, once the heat-killed smooth strain and the normal rough strain, when injected separately, they are harmless. They cannot kill the mice. But something interesting happens when both these are mixed together. That is, the smooth heat-kill strain and the rough strain are mixed together. They should have ideally not killed the mice, but Griffith observed that the mice dies. So from this observation came the idea of transforming principle, that something in this mixture is there which makes the rough strain, which is the live strain, virulent. And this virulence factor definitely has to come from the heat-killed smooth strain, which otherwise, if alive, causes the disease and kills the mice. So this is basically the transforming principle, which was put forward by Griffith. Later on, Oswald and Avery, you can see in the other panel, set up some experiments to prove that this transforming principle is nothing but DNA. So what he did, he took heat-killed smooth cells, as you can see in this picture, and he added proteinase. They added proteinase there. So proteinase will destroy all the protein molecules. So that means if protein is the disease-causing factor, then we can see that the rough cells should be transforming into smooth cells because it still has DNA and RNA in it. So adding proteinase takes care that the protein is totally destroyed while the nucleic acids are left intact. So here, either the DNA or the RNA is the transforming principle from experimental number 1, where the protein is added. It was proved. To rule out whether this is DNA or RNA, in the next experiment, they added RNAs, and RNAs destroyed the RNA in the extract mixture, and they added the R cells. So these heat-killed S cells, with the addition of R cells, live R cells, got transformed into live S cells. So the only factor that remained, transforming principle that remained to be a potential candidate is DNA. So when they finally added DNase, they found out that if DNase is added, the DNA is destroyed. And since DNA is destroyed, so DNA no longer can transform the hit kill S cells, and that is why in this last case they did not see any appearance of the S cells. So these confirmed that the transforming principle is DNA, ruling out the possibility of protein and RNA as the factors which causes the transformation. So I'm not sure whether madhuammu600@gmail is listening to this discussion, but if she logs in later to the live recording of the live session, I think her query she can find out in the recording. So in the meantime, let me ask my technical support team to find out if somebody has raised some queries. There are no any live queries as of now. So let me go to the second query discussions, which was by Gargi Joshi, 111@gmail.com. Gargi had a question in the video at 23.12, and she was asking that she do not understand the concept of operons, and if we can explain it in a better way or in a different way. So we have briefly already discussed about operons, but here let me once again elaborate the discussion on operons. It is known to many of you already that operon is a functional unit of genetic regulation, which is found in low organisms like bacteria and archaea. So in such organisms, structural proteins with related functions are usually encoded together within a region of the genome in a block called an operon. So basically here, all these proteins or would be factors or some, catalytic enzyme which participate in a common metabolic pathway. So basically, a operon consists of promoter. As you can see here, there is a promoter and an operator and a group of structural genes. So this is, you have promoter, operator, and structural genes. Together they constitute one operon, and this entire operon is under the regulation of a single regulatory element or regulatory gene. So when one gene is expressed, which is under this particular regulatory gene, automatically the remaining genes following it will also be expressed. So when A is transcribed, along with it B, C, D will be transcribed, and they will also be translated together, resulting in the production of protein A, B, C, D. And all these four proteins would actually participate in a common metabolic pathway. So now we know that operons are transcribed together under the control of a single promoter, as you can see in this figure, and there is results in the formation of a polycysteinic transcript. Thus, for the activation and deactivation of a particular metabolic pathway, the regulation of the transcription of all the structural genes encoding the enzyme that catalyze the various steps in a single biochemical pathway can be controlled simultaneously as per need. So if there is no any need for B, A, C, D, producing A, C, D is also not required. So if there is a requirement for the product protein A, we would require the protein B, C, D, because only then the whole metabolic pathway would be completed. So this is one of the economic reason why all these genes which participate in a single metabolic pathway are put under one single regulatory gene, so that they are easily transcribed and translated whenever the need arises. So let me move to the third query, which came in the online classes. This was asked by Omid Costly 74. He was asking reason for pea plant used in Mendel experiments. So these are all known to many of you. Let me put up briefly. Particularly in breeding experiments, we want organisms which have short life cycle, so that we can produce more generations, progenies. Because to see and observe heredity, if we take crops or organisms with short life cycle, we can complete our breeding experiments rapidly. So that is exactly one of the reasons, fast growth, which allowed for multiple generations to be studied in a relatively short amount of time. Mendel took pea as a model plant. Another thing is, whenever we are doing experiments with organisms, plants are sometimes better, easy to raise and maintain. So this is one more reason why Mendel took pea. And many things about pea, it's particularly its agronomy was known, how to grow. So all these made it easy for raising the plants and maintaining them easier. Since he was looking into heredity, so he was looking into phenotypes or features or characteristics which are easily visible. Pea plants offered Mendel several visible characteristics, such as flower color, seed shape, plant height, or even wrinkled or round seed shape. So these characteristics were easily observed in his study. So the documentation also became quite easy. In a breeding experiment, having control on the breeding is very, very important. So the fourth reason why pea was chosen was controlled breeding. These pea plants can be easily crossbred and also self-fertilized, allowing for various controlled breeding experiments. Another point due to which pea found favor with Mendel was that the purity of traits. He was able to establish pure breeding lines of pea plants, which always produce offspring identical to the parent, allowing for precise observations of inheritance pattern. Let me once more check if there is any online query. So there is no any online query. So NPTEL team also let me know if whenever there is some online query, so that I can respond. So there was a query by Devansi Sangvi 986@gmail.com. So she had a question at video 33.01. Is cloning site the same as restriction site? The answer is true. So if we want to clone something in a vector, we have to cut it open, which is the restriction site. And in this site itself, we will insert our DNA of interest. So when we are cutting at site A, our insertion site is A itself. So restriction site and cloning site is similar. Sometimes we use double restriction digestion. So we may cut at two sites A and B. So this helps us in kind of directional cloning. So here also the restriction sites between A and B would be the cloning site. So I think this is very simple question for those who have already understood. But sometimes it may happen that we may have a restriction site at A, and into that we put a stuffer region or DNA. Stuffer region or DNA. So now this stuffer region, or kind of maybe an adapter also, or so these may itself have some multiple cloning site inside, right? So, and these restriction site may be different from restriction site A or restriction site B. So in this case, although the site of restriction and the site of insert hello, yes, sir. Volume is there. This is in which, this is which chapter you can do. Okay. Am I audible?
Yes, sir. Yes, sir. You're audible.
Yes, sir.
There is another question by Devangshi. Devangshi Sanvi 986 gmail.com. So she is asking, how do cloning vectors express themselves? If not, then what is the point use of them? I think this is a little bit misplaced question. Cloning vectors cannot express themselves. Vectors can only replicate. So I'm not sure what she is asking. Probably she is asking about the expression of the DNA fragment or some gene that we have inserted in a cloning vector. So for those who require a little bit of detailed, about all these basic techniques, I have a course in NPTEL on genetic engineering. Kindly please go through that course, which will be very helpful in understanding the basics of genetic engineering, which is important for gene editing. Anyway, let me add to the query by Devanshi Sangvi, who is asking, how do cloning vectors express themselves? Cloning vectors do not express themselves. Cloning vectors will multiply or they will replicate n number of times so that they produce more clones, more copies of the same piece of DNA that we are interested in. For expressing that particular target DNA or gene, we have to use another vector called expression vector. So there is a difference between cloning vector and expression vector. Let me go to another question by Devanshi Sangvi 986, who is again asking, do cloning, expression, and subtle vectors come under plasmid, or is plasmid a cloning vector? Anyway, so these are all basic concepts which are available in genetic engineering textbooks. But since Devangshi maybe not having a foundation in genetic engineering, let me tell her what is a cloning vector. and what does this cloning factor contain? So if you look into a cloning factor, physically it is composed of a single piece of DNA from a single organism, or from many multiple organisms, and which may be taken from virus and or some bacteria or the cell of a higher organism. And basically these cloning factor characteristics that it can be stably maintained in an organism and into which foreign DNA fragment can be inserted for multiplication purpose or cloning purpose. So basically we use the vector DNA to carry foreign DNA fragment into the host cell. Through these, we obtain multiple copies of the foreign DNA fragment, and these cloning vectors will have an origin of replication, then restriction site, and a selectable marker. It can be plasmids, cosmids, or bacteriophages, or having elements of some of these DNA molecules. And since they are having origin of replication, they are self-replicative, which is well understood. And there are various features which make a cloning vector ideal. We already told about origin, the origin of replication, which is important. Without this origin of replication, it will not be self-replicative. We refer to antibiotic resistance, which helps in the selection process, and also the restriction site, multiple restriction sites. So these multiple restriction sites helps us or gives us the freedom to work with any available restriction enzyme in the laboratory. Now we have another type of vector which is called as a shuttle vector. So this shuttle vector is like a shuttle bus which travel from one point to another point, carrying passengers in between the 2 points. So here, the shuttle vectors can replicate in more than one organism, one type of organism. So it may replicate in E. coli, which is a prokaryote, and it can also replicate in yeast, which is another type of organism. So in order to be able to do this, such a vector must have certain essential elements. Particularly, it will have two origins of replication. As you can see, here is a origin and replication of genes for bacteria. So this is one. Other is the yeast origin of replication. So this will come into action when the vector is inside the bacteria, and this will come into action only when it is inside the yeast. So it has at least two origin of replication, one each for each type of organism. It will have a yeast centromere sequence so that it is partitioned into daughter cells during yeast replication, and it will have selectable markers for both yeast and E. coli and multiple cloning sites for inserting the gene of interest. So this is the makeup of a subtle vector, and I think with these we come to end on the discussion of subtle vectors. Let us now go to the next query. This query is by S. Pandey 508@gmail.com. He's writing, "Hi, I have a question about what is happening in this video 2514, but I think he has referred to the wrong timeline in the video. His question is, he's writing in books, especially in Genome's tree by T.A. Brown, genome is identified as total genetic content of the cell, which includes two sets of genes and mitochondrial genes in human. However, in your slides, genome is defined as the haploid set of chromosome in a diploid organism. So what is the true definition?" I think there is a misconception here. So as I track down, these are the slides probably S. Pandey is referring to. This slide time 3.08. So here I have given a historical reference about the term genome, who introduced it first. Here we are not trying to giving the definition of genome and what is the total content. We are not trying to convey that information here. Here we are trying to convey about the historical development of the concept of genome. So this term, as you can see in this material, which is also available in the MOOC's website, you can download. This term was introduced in, the term genome was introduced in 1920 by the German botanist Hans Winkler to describe the haploid chromosome set. So this is not my statement. This is the statement attributed to German botanist Hans Winkler. So he used this term genome to describe the haploid chromosome set, which together with the pertinent protoplasm specifies the material foundation of the species. So we have now later on added here, we humans are diploid in nature. The half set of the diploid chromosome are what we call as the haploid chromosome set. We are not telling this is the full human genome. So if you go to the next slide at 3.48, this is what we have stated. The genome is a complete set of genetic information in an organism, not haploid. We have not stated anywhere that it is the haploid. So it is the complete set of genetic information in an organism. There is no doubt about it. It provides all of the information the organism requires to function in living organisms. The genome is stored in a long molecule of DNA, and we all call them as chromosomes. In eukaryotes, each cell genome is contained within a membrane-bound structure called the nucleus, so on and so forth. In addition to these, let me also inform that total genome is the nuclear genome as well as the mitochondrial genome in case of animals. However, in the case of plants, it has a third genome, which is the chloroplast genome. Okay, so this has to be very, very carefully read at this point of time. Now, referring back to the introduction of the term genome by German botanist Hans Winkler, you can refer to these literature in the journal Genome, volume 62, number 10. So the concept of genome after 1 century of usage. So here you will come to know that Hans Winkler introduced the term genome. At that point of time, he described it as the haploid chromosome set, which together with the pertinent protoplasm specifies the material foundation of the species. So this is, we are taking, we are quoting verbatim exactly what Hans Winkler was telling while introducing the concept of genome. But as in the case of many scientific ideas and terminology, the concept of genome has undergone huge change. So if you read this article, the concept of genome after 1 century of usage, you can see how the concept has gradually evolved from time to time. There is another question by Devansi Sangvi 986. So she is asking again a very fundamental question: what are titers? So she was referring to what is happening in this video 1710, and she is asking what are titers, and it is all known to you, many of you, all of you maybe. Generally, this is a measurement of the amount of concentration of a substance in a solution, and refer to the amount of maybe some kind of biomolecule in a person's blood. It may be some kind of antibodies, for example. Okay, so with this, we come to the last question by Bala BioNano 95@gmail.com. So Bala BioNano 95 is asking what is happening in this video at 22:46. So I can't understand the E. coli Orion deep part in arrangements. Is that part easy to find out the restriction enzyme for to cut specific genes or its structural form gene. Okay, anyway, let me go back to this once again. So for in this plasmid, as in the case of this plasmid, which is a origin of replication, in bacterial genome also there is a origin of replication from which the replication will start and proceed in both the directions, left and right. And as the replication goes in both the directions, left and right, both the replication front go and meet at certain point, which may ideally be exactly the opposite point, equal from both the equal from ODC in both the arms, but sometimes it may not happen. Of course, one arm may have rapid replication. So now DIF is basically the short form for deletion induced filamentation. This is a specific DNA sequence which is located near the terminus region of the E. coli chromosome, and this site is involved in chromosomal deconcatenation and is a preferential site for exogenous DNA integration, contributing to genome evolution, also virulence gene acquisition. So I think with this we have come to end with the queries put forward by various students. Let me now check if there are any. Okay, there is one. Sir, can you explain the difference between anase and alternase and homologous? Subronil Saha is asking this question. So, okay, let me go to this is probably you were asking from lecture 1. Right. Okay, so I think this is visible there. Oh, so it is visible, right? So you are asking about non-homologous end joining and homologous recombination, and then the alt form of the NHEJ. Okay, so here you can see these are actually repair mechanisms. Whenever there is a strand breakage in DNA, this kind of repair mechanism comes into action. So in the case of homologous recombination, you'll see a double strand breakpoint is present here in one chromatid, and this is repaired using its sister intact sister chromatid as a template. Okay, so there is repair of breaks by homologous recombination, therefore is a highly high fidelity process, and it ensures that all the genetic information at the break site is retained. So here you always have a template against which the repair is taking place. So it is based, that template provides the homology for this kind of repair. But in the case of non-homologous, in the case of non-homologous repair, so what is happening is simple rejoining of the broken ends, regardless of the DNA sequence. So here we do not have a template. So wherever there is some kind of DNA breakage, so you have certain proteins which will come into action at that point, and they will try to fill up this gap. So since there is no any template here, so there is the repair process is completely random and not homology based. So it may incorporate any kind of base in this space. So therefore, this will actually give rise to error-prone repairing, and this also therefore gives rise to mutation. So these are the two main differences between the two homologous recombination and non-homologous recombination. So the third form is the alternative form of the non-homologous and joining. So here the difference, you can see that there is a short range resection, and due to this afterwards there's an alignment of some homologous sequences over here, and there is a flap removal and ligation of the DNA ends. So you can clearly see here the repairing mechanism and also the factors which are involved in each pathway are different. So this is in brief the differences between homologous, non-homologous, and alternative NS-EJ. Okay, now there is another. Let me go and check out. There is another query. Another query. Probably this query came in from YouTube. Gokul is asking. What Gokul is asking? Is it possible to edit human DNA to cure genetic disorder? The answer is yes, it is possible. And in fact, one experiment was already done in China where they were trying to create babies who are resistant to HIV. Okay, so I think in one of the future lectures we will be having discussion on that. But the question is, we are yet technology is ready, so that has been actually proven. In animals, experimental animals, we are already doing it, and by mistake, it was done in China, but ethically this is not allowed. We have to remember that. So these are things we will discuss again in the future lectures. But to be brief, is it possible to edit human DNA to cure genetic disorder? The answer is yes, but this is not allowed by human and medical ethics. Any other queries? Any other queries? So there are no any queries as I can see in YouTube. Find out. Yes. In YouTube also it is not there. Sampai ground dulu kok. Okay, since there are no any further queries, we can wind up. So if NPTEL team give us the feedback.
Yes, sir. We can wind up the session.
We can wind up the session. Okay, let me thank my technical team. So sorry, there is a echo from the right. So since there is no any further queries, let me wind up by thanking NPTEL support team in CET and also my technical team here, Mr. Pulok Basumatari and Udangsri Bora Sakya Bora and John Jyoti Kalita. Thank you.